Hey everyone, I am going to explain what is the platelet satellitism or rare cause of pseudothrombocytopenia. Thrombocytopenia means decrease the platelet count. The normal range of platelet is 1.5 to 4.5 lakhs. So pseudothrombocytopenia is patient is having adequate platelets in the range of more than 1.5 lakhs, but the CBC cell counter showing decreased platelet count less than 1.5 lakhs. What is the exact mechanism in the satellitism? What is the platelet satellitism? Why it is called pseudothrombocytopenia? Why CBC cell counter not able to measure the platelets? So the because there is a glycoprotein second B and third A complex on the platelet membrane, and they, when we are using EDTA bulb for sample collection for CBC, so there is a formation of antibodies in not all the patient, but in the some patient there is a formation of antibodies against this the glycoprotein on the platelets. So the platelets are directly stick to the WBC surface. It may be stick neutrophil, lymphocyte or monocyte. Okay, so suppose this is the platelet. This is the tiny platelets having the receptor glycoprotein second or third receptor. So there is formation of antibodies against this receptor. So there is a formation of antibodies and antigen complex. Okay, and this complex stick to the white blood cells. Suppose this is the neutrophil, this is the one lobe, second lobe and third lobe. So platelets are stick to the neutrophil surface, so in the satellite fashion. That's why it is called platelet satellitism. So basically CBC cell counter having two chambers. Okay, first one is RBC chamber plus platelet chamber, and the second one is the WBC chamber plus hemoglobin chamber. Okay, so if the platelets are directly stick to the white blood cell surface, in spite of going into RBC platelet chamber, they directly going into the WBC and the HB chamber. So there is a no mechanism to count the platelet in WBC and HB chamber, because platelets are always measured in RBC and the platelet chamber in 3 parts or 5 parts of CBC cell counter. So that's why the CBC cell counter unable to count the platelets, because the platelets are not going into the platelet chambers, because they are directly stick to the white blood cell surface, either neutrophil, monocyte, eosinophil, any cell surface, any white cell surface, because there is a formation of antigen antibody complex. Antibodies are directly against the glycoprotein second and third, which is of the platelet membrane. So this is the EDTA bulb which are using for collection of sample, generally for CBC. Okay, so come to the peripheral smear examination. So how to resolve these problems? Always do peripheral smear examination. In the peripheral smear examination, you will get the platelets around the white blood cells. So this is the one cell showing the platelets around in the satellite fashion. Now come to the high power field. So this is the one white blood cells, and you will see many platelets are directly stick to the surface of that white blood cells. So that's why we are called it's a satellitism. Now this picture is more clear. Lymphocytes and the tiny dots is the platelets. They are directly attached to the surface, or they are scattered around the white blood cells. This is the another picture, and you can clearly see that there is a clump of platelets. So these clumps are not directly measured in the CBC cell counter, and you will always get pseudothrombocytopenia on the CBC cell counter. This one is a very nice picture. Here you can see two lymphocytes and one lymphocyte showing collection of the platelets around the lymphocytes. Thank you.